thank you very much. I'm the CEO of PredxBio. What a great day today, right? We started off with Nubar talking about polyintelligence. And then Najat Khan talked about in silico clinical trials. And now Pooja talked about computational pathology and how important that's going to be ultimately in determining the mechanism of action. So that's what we do at PredxBio. We try to understand the mechanism of action using spatial biology. So how many of you have heard of spatial biology? How many of you actually have heard the term? Show of hands. Okay, around 15%. So spatial biology is really about understanding the relationship of different cells in the tumor microenvironment with respect to the interactions, with respect to how they conspire together to move the disease forward, and really getting to the mechanistic underpinnings of the tumor microenvironment. Eric Topol calls it today, arguably, the most important technique to understand tumor biology. And that's what we've been doing in PredxBio now for 5 years. Very pleased to see Nature Methods actually call this the method of the year. And Nature Journal actually called this, actually have a full journal devoted to spatial biology recently. Eric Topol talks about spatial medicine, something that's going to become more and more important in the very near future. Medicines based out of spatial biology, which is understanding how cells interact with each other and how they signal to each other. And this is kind of what Pooja was referring to when she was talking about computational biology or computational pathology. Chakra Chandabotla, who's here, who's the founder of the company, actually coined the term spatial biology around 5 years ago. And today there are 2,500 papers talking about spatial biology. And we have published around 25 publications on this. Again, as I said, Nature Methods just called us the method of the year using spatial proteomics. So what is the biggest issue that we first started tackling around 5 years ago? The last panel talked about this. Immunotherapy was growing at an amazing pace, right? I mean, every year, around 30% increase in immunotherapy clinical trials. But the inconvenient truth is that the response rate for all the drugs that the panel was developing in the last 10 years has been in the 20 to 30% rate. So it's not really changed over the last 10 years. And we really believe the reason for that is lack of understanding of the hidden circuitries of the tumor microenvironment. If you look at the PD-L1 biomarker that was discussed in the last panel, 30% of the patients are selected on the basis of PD-L1 biomarkers. And when you look at the response of these patients actually who are PD-L1 positive, 5 to 40% of them have hyperprogression, 20% of them have severe adverse effects, 30% of them, kids really, have hyperprogression, and sometimes, lo and behold, PD-L1 expression negative patients respond to therapy. That is the real issue right now with respect to IOs. And that is also the reason, the lack of a reasonable biomarker, why 90% of these clinical trials that were just being discussed are failing today. It takes 10 years, a couple of $1 billion to develop a drug. Spatial biomarkers can be game-changing. What we have seen is that instead of 30% success rate or less in clinical trials, with spatial biomarkers, you can actually go up to 90% success rate in clinical trials. And we have seen it working with pharma companies over the last 5 years. And the way we do it is really separating the responders from the non-responders using the spatial signatures of the tumor microenvironment. We'll talk about this in a very short time. What we see is some more like what you see on the right-hand side, responders versus non-responders separated out. This leads to better stratification and selection, much better drug efficacy, much better companion diagnostic tests, and really the dream of personalized medicine coming true. We also do this in phase 1, not wait till phase 3 for the failures that you see in the clinical trials in the pharma companies, but start in phase 1, differentiating responders from non-responders and revealing the mechanisms of action. Mechanism of action is all about the why behind why a patient is responding or a patient cohort is responding and why a patient cohort is not responding. Once you have a successful phase 1 trial, you can actually have a very, very successful phase 3 trial, is what we have seen working for the last 5 years with pharma companies. And we have been doing it not only across IO, but also ADCs, multispecifics that were being discussed. These are some of the results that we have seen till now. PD-L1 expression-based test, the area under the curve is around 0.6. What we have is 90% confidence in terms of response to immunotherapy in our predictions. We have also looked at combinations of immunotherapy and ADCs, which were discussed in the last panel. Today, the response rate of most ADCs is in the 30% range. When you combine with IOs, it goes up a little bit. And the reason for this is the lack of understanding, again, of the tumor microenvironment, the relationship and the interactions between the bystander effects in the ADCs and the immunotherapy agents. And again, guess which biomarkers you use today for these clinical trials. There are at least like 20 clinical trials running with ADCs and IO combinations. Again, it's the same old PD-L1 expression, which is essentially giving the same response rate for positive, PD-L1 expression positive versus negative. So that is the challenge right now. The pharma companies are slow to change from the traditional biomarkers they've had, and now they're extending it to IOs and ADCs. So this is what we do. We reveal cancer biology via actionable AI-driven spatial biomarkers. 10 years ago, we had the realization that heterogeneity is not random in the tumor microenvironment. There is an organizing principle to heterogeneity. That heterogeneity can be expressed via the microdomains, which we'll talk about, that we discover in the tumor microenvironment. Once we understand the microdomains, we are able to reveal the mechanism of action. So with our technology, we unlock the intratumor heterogeneity. We try to develop actionable biomarkers and really power clinical trials, enriching patient populations to achieve a 90+% accuracy in predicting patient outcomes. So we start off with a tissue biopsy. We are able to label the tissue cells with certain proteomic biomarkers. And once we do that, we take a digital image of that, and then the magic starts. So this is the tumor microenvironment in all its complexity. Tumor cells, stromal cells, immune cells. We are able to unravel the cell-cell communication based on the ligand exchanges that we are able to capture based on the signals, and we are able to capture these microdomains. These microdomains are constellations of cells coming together in a specific spatial configuration. And we're able to see that these microdomains also repeat themselves with the computer vision and the AI technologies that we have developed. Once we capture these microdomains, we are able to correlate these microdomains, the spatial signatures of the tumor microenvironment, directly to the response. We believe that the microdomains represent some kind of a fundamental evolutionary block of cancer. Now, once we do that, with the AI system being embedded with true explainability, we can expose emergent biology, the mechanistic underpinnings of cancer, pathway crosstalk, and also identify hybrid cells, cells that are actually going from one state to the other, mesenchymal to epithelial, for example. We have published this in 25+ publications, heavily patented our technology, and let me show you the kind of the SpaceIQ engine that actually does this. So data from spatial proteomics, spatial transcriptomics, bright field pathology can be inputted into the SpaceIQ engine. And after some very basic quality control, we are able to phenotype every single cell of the million cells on the tissue slide using this recursive tree and leaf algorithm on the right-hand side. Once we know the spatial location of each of these cells, who's next to whom, who's talking to whom, what kind of signals are going on, we're able to capture from that these microdomains, which are repeatable constellations of cells coming together in a specific spatial orientation. Once we capture the microdomains, we're able to expose the network biology or the mechanism of action. It's been very helpful for us to do that over the last 5 years with a number of clinical trials, and the technology that we use has moved away from the black box deep learning that 99.99% of the companies and the pharma companies are using today to really leverage Bayesian network inference, spatial parametric models, and pointwise mutual information to understand how cells are interacting with each other and get to a point where we can actually find these transition cells, which are cells in transition, and we have found these cells in transition are pivotal in terms of the prediction of response to therapy. So you got to understand how cells are going from one type to another, in this case, the epithelial to the mesenchymal cells. We play in discovery for multiplex spatial omics, where we actually use a multiplex biomarker to expose the mechanism of action, enrich the patient in phase 1 and 2, and then move to phase 3, where we can actually deplex a high-plex panel and maybe use just a combination of IHC and a couple of IHCs and H&E to be deployable across all the sites in phase 3 with a similar degree of accuracy, 90% predictive power across the board. And the reason we can do that is the explainability that's embedded in our AI algorithm, as well as the unbiased way in which we find these microdomains. So what we have done over the last few years, just a few quick examples of what we have done: predicted patient response with over 90% accuracy in a number of different cancers, including colorectal, diffuse large B-cell lymphoma, non-small cell cancer, etc. We've also revealed the network biology early in the trials to actually get a companion diagnostic test with very high accuracy. And then, we are very good in understanding from the spatial biology some of the more ambiguous situations when you have atypical ductal dysplasia, atypical ductal hyperplasia, where to understand if there is the tissue is malignant or cancer. This is where a lot of dilemma among pathologists lie. And also take virtually multiplexed images from transmitted light and combination of transmitted light and IHC. Some use cases. This is for diffuse large B-cell lymphoma. The challenge here was to identify responders versus non-responders in immunotherapy clinical trials. This was done for a medium-sized pharma company. We had accuracy of 99% with our SpaceIQ platform in predicting patient response to therapy. In this case, it was a bispecific immunotherapy antibody. So this is basically what we saw in terms of complete response versus progression of disease using an 8-plex system. On the left side, you have complete response. On the right side, progression of disease. We have done this for colorectal cancer, another use case that we can show. We were able to predict progression of colorectal cancer with 93% accuracy in a 400-plus patient cohort. So again, this basically shows the jump that we had from biased biomarker intensities and genomic tests all the way. You can see this on the graph in the middle, 93% accuracy in predicting colorectal cancer progression. Not only were we able to get these kinds of accuracies, we were also able to expose the network biology of cancer, which was extremely critical for the pharma companies that worked with us. On the right, you see the network biology for recurrence within 3 years versus no evidence of disease for 8 years. And again, these were done from the microdomains that you see here, no evidence versus recurrence. In lung cancer, we have looked at non-small cell lung cancer extensively, and what we have been able to do is identify responders versus non-responders based on spatial combination of metabolic biomarkers. We can talk more about this at the reception, potentially, where we have been able to predict with 90% accuracy the response to, in this case, immunotherapy. And not only that, we've been able to expose the underlying metabolic programs that basically define the mechanisms of response. Not only have we been able to predict response, but also survival to ICI or immunotherapy treatments, 92% accuracy in predicting survival across different stages, and also expose the underlying mechanisms of resistance. Again, all of this were done from microdomains like this with a 40-plex system. This is where we have been able to reveal the microdomains of response and resistance. Here, the various lines indicate the interactions between different cells. The thickness of the line represents the strength of those interactions, and then the different colors represent what's more prevalent in responders versus non-responders. Again, this shows the spatially and functionally distinct catabolic and anabolic programs predicting response in non-small cell lung cancer. So we've done this over a number of different cancers. This last use case is really to show that we are able to take a high-plex system in phase 1, 40 to 60-plex system, and then essentially deplex it when we have a CDX or even a phase 3 trial to get to a much simpler system without losing the accuracies of prediction. And you can only have this when you do the whole study in an unbiased way, and you have explainability embedded across the AI algorithms. We've done this for a number of different cancers, from colorectal to lung to glioblastoma to lymphoma. And our dream is to have a new drug that we can actually match with a patient's signature and be able to predict using the SpaceIQ engine 90% accuracy for patients who benefit versus don't benefit, all based on their predicted, based on their spatial signatures, microdomain 1 versus microdomain 2 in terms of responders versus non-responders. We have just started taking this beyond oncology to neurology as well as dermatology, and going beyond immunotherapy to ADCs and ADC and IO combos, and also from a spatial technology going beyond proteomics and transcriptomics, going to spatial microbiome and spatial metabolome and spatial epigenome. You know, today the discussion started with Nubar talking about polyintelligence, and and that polyintelligence really is related to what we are doing in terms of understanding the tumor microenvironment from a spatial perspective, from a genomic perspective, from a proteomics perspective, from a perspective of really understanding the underlying mechanisms of resistance and the underlying mechanisms of response. So we call this spatial intelligence or spatial awareness. And so from where we started 30 years ago, where disease was defined by the tissue of origin, to now disease defined by tissue and mutation, to the future where disease is going to be defined by spatial multi-omic signatures. That's kind of where we want to go from here, and that's where we've already taken a number of pharma companies that have worked with us. So if you want to really take a look at the unbelievable views of the tumor microenvironment, we have an Apple Vision Pro that we'll have at the reception. You can stop by and take a look at it and really enjoy how these microdomains come together and how the constellations actually predict, exposing the network biology, the response to no response. And we really believe that this is the future of where oncology is going, and spatial intelligence is going to get us closer to the promised land. So thank you very much.